All right, we're gonna get started today. Just a few housekeeping things. At the end of the session, there'll be a raffle for the 2 gift cards, so just keep your tickets handy. If you have to leave to go back to your bench or whatever, you can still claim your prize later. And just a reminder for Friday, there's a celebration in De Gasperis, so there'll be food, coffee, drinks, and it's from 11:30 to 1:30, I believe. And so for today, we have 2 talks. So I'm going to introduce our first speaker. It's Shabnam Salehi-Rad. And she's been part of the Cytogenetics lab at UHN for 8 years. And she has a background in both cytogenetics and molecular, and additionally has experience with research at U of T, but she prefers the diagnostic environment. And so the title of her talk is Going Digital. So take it away.
Thanks. Hey! Hi everyone. As I was introduced, my name is Shab, and thanks for having me here, first of all. Okay. And I'm going to do a very short presentation on going digital in the cytogenetics department here, or cytogenetics lab at Toronto General Hospital. So does anyone actually know what we do in cytogenetics? It's always the case anyway. So the cytogenetics lab, cytogenetics in general, it's a discipline that looks at chromosome patterns in order to see abnormalities within them. And so basically chromosome analysis or karyotyping is the analysis of metaphases which have been banded using trypsin and then stained using either Leishman or Giemza method. And this will create unique patterns. So as an example, there you go. Over there you'll see a metaphase, and this is basically, it's captured for us. So we use a system that scans slides, stains slides, and captures these metaphases for us to look at a later date on the computer. And we also use software called Icarus in order to convert that metaphase into a karyotype. So basically this allows us to align each chromosome set and look at the unique patterns to see any abnormalities that might be present. Another thing that we actually do in cytogenetics, another test, would be FISH, which stands for fluorescent in situ hybridization. And so we use it on metaphases and interphases and on different sample types like bloods, bone marrows, in situ paraffin embedded tissue. And basically what FISH allows us to do is a fluorescently labeled probe is annealed to a specific region on a specific chromosome. So it gives you a better detailed look at a specific chromosome versus a karyotype. So for example, in this one here, this one, let's say that's chromosome 8, and the green probe, green region is a control. So both the chromosome 8s have the control, but one of them is missing the red. So there's some sort of a deletion. That's an example of one of the tests that we do. So some facts that I want to give you about our lab. I mean, I guess in comparison to the molecular lab, we do quite a less samples. But in 2018, we got around 4,900 samples. But not all of these samples are actually activated. So all samples, they go through logging in, accessioning, and basically culturing and harvesting. But only the activated ones will go further in terms of slide making, scanning, capturing, and analysis at the end. So about 3,200 of these cases were activated, but in total we did 4,800 tests. And these tests can be a combination of cytogenetics, karyotyping, as well as FISH. So a case can come in for FISH, but there are actually 3 probes that we are looking at, not just one. So anyway, on a yearly basis, we also use hopefully we reduced it, quite a bit of paper. I would have to say for a normal cytogenetics case, we would probably have around 13 to 15 pieces of paper that would be in an envelope, and then the director would get that case and sign it out at the end to the physician, the results, I mean. And we also had to worry about storage fees because all this information has to be stored somewhere, right? So we had to pay storage fees on a yearly basis as well. So going digital really, or using the software that we already had to its utmost, allowed us to not only reduce paper, go all environmentally friendly, but also save a little bit of money, aside from allowing all the information for a specific patient to be available in one place in a secure fashion. So these are the areas we tried to go digital on, and I'm going to go into each one of them, tell you how we used to do it before and what we tried to do, what changes we made in order to go digital, basically. So when a sample comes into our lab, what happens, the flow of the work is each sample is given a unique login number, cytogenetics number, and then all the details of the sample are also written down. So what we used to previously have was a login binder, and a technician would give a specific unique number and then write the details of the sample, like first name, last name of the patient, when it was received, type of sample received. So instead of doing this manually, like writing it down, we tried to go completely digital by copying that login manual into a Excel format. So now the technicians can just go into the login book digitally, enter all the data, and then they'll do that on one monitor. On the next monitor, they'll have CoPath open, and they can just directly enter all the necessary data for that sample into CoPath. And this also allows us to, at the end of the year, before we used to actually scan all the login binders into the system, because we have to keep that information, and we don't have to do that anymore. It's just directly in there. It's secure because it's password protected, and it's also recoverable because it's on the UHN network. So no loss of data, basically. So next area where we tried to go digital was the analysis and the checking of metaphases and karyotypes. The analysis portion was really already digitized, right? Because we use metasystems basically to capture images and allow us to analyze later on. So, but we also have to use another software called MetaClient, and the only thing that we did with MetaClient was basically print out an analysis sheet which would have details of the case. For example, 20 cells analyzed, which cells were analyzed, what the results for each metaphase was. So we thought, why not just use metaphase for all of that? There's no need to print the analysis sheet, the images, so no more paperwork. So you can see in MetaClient right now, you can see all the images that were analyzed on ICRIS already in MetaClient. So if you need to, you can actually click on each one of these and open them up. No need to, go back and forth kind of thing. And also, you can use MetaClient in order to see a little bit more detail for each metaphase, really. So you can see the results for each metaphase, which cell it is, which slide it's on, and which culture it came from. And so the analyst would go through a case, analyze a minimum of 20 cells, if they have 20 cells, and then also indicate to the person checking the case which metaphases they should look at, because a checker can't look at all the metaphases, essentially. So that's the category right there to check. So this indicates to the checker, these are the 4 that I'm looking at. So once the case is passed on to the checker, they'll look at those 4 cells, and they can do that by combining the karyotypes. So they'll look at all the karyotypes at once instead of looking at paper images of each karyotype one at a time. So they can go ahead and compare chromosome 1, chromosome 2, and catch, look at the abnormalities that were found in the case. In addition, the beauty of MetaClient is the fact that you can keep track of where a case is at. So you can, right here, there's a bar that basically shows you when the analysis was started and by whom, and when it was completed and checked, and when the director actually signed out the case. So an analyst, when they start the analysis, will move the bar to the analysis started, and that will stamp their name and the date they started the case. The same goes on for the checker. And then once the director gets the case and signs out their report, they will move this bar to checked, and that will stamp their name and the date they did that. And the case will be locked, so no more changes will occur to the case unless we really have to. So that's the part for karyotyping. And also, I skipped okay, so and entering and checking for fish was the same thing. We would actually print the score sheet. So the scoring would be completed, and the score sheet would be printed out and used to enter the case into COPATH. So why do that? Just use the computer. You can always bring up this score sheet. You can enter all the data into COPATH, and then the checker would come and check everything that was entered into COPATH and just attach this score sheet into MetaClient. So all the data, all the images, all the cells analyzed, all the back matter in terms of external requisitions were all in one software, so, and easily accessible. So really the advantages of all of this was very little paper usage now, maybe 1 to 4 or 5 papers per, not even? Okay, great. And the most advantageous part was the fact that now we have one area that we go to. We just have to click, go into MetaClient, and there's all the information for that case. There's no more going into, oh, let's find the envelope with the images in there and the analysis sheet that's over there. It's all in one place. It's secure. It is recoverable. Each individual technologist actually has their own login and password, so when they go into a case, we actually know who went in there, what did they do in that case. So all the changes are basically recorded within the system. So, and of course, we're being environmentally friendly with less paper usage. And the most important thing for our directors was the remote access part. So our directors now can be anywhere in the world as long as they have access to the UHN system. They can log into MetaClient and they can view cases and sign them out, which is ideal for a patient. You know, if they're waiting for results, they will get it. No delays really. And in addition, they can also keep track of the types of cases and where a case is at. So is it in the analysis area? Has it been checked, and will it be received by them soon, just by using MetaClient? Finally, in the long run, we're going to use MetaClient in order to keep track of what kind of case tests are we actually doing. Let's say someone is asking us, how many AML cases did you do last year? So we can just do a simple search on MetaClient and come up with, I don't know, let's say 500 cases of AML, or how many multiple myeloma cases, the same deal. So in the long term, there's quite a bit of savings, and also making sure that the system is secure and easily available anywhere, as long as it's password protected. So there's no, unwanted individuals getting into the system, basically. So hopefully that's a little bit of a kind of quick view of what we've done. And if you have any questions, let me know. I just want to thank the cytogenetics department for being so supportive and so, yeah, right there. And that's my presentation. Thank you very much.
Are there any questions for Shad? Chris?
Just a general question. Would those results then go back to the electronic patient record at UHN for cytogenetics?
The records, I think the final report should be available, but I'll be honest with you, I don't know much about the electronic patient. Yeah, so I don't know.
No, they're actually transcribed, like they're taken by the cytogenetics system and the procedural report, which then gets reported out.
Oh,
Yeah.
okay.
So because they're not digitized.
Any other questions? All right,
Thank
you
you.
very much. So we have one more talk for you guys today. The talk will be presented by, The second speaker is Sherry, and she works in the cytogenetics department as well. Sherry has been an MLT for over 25 years, working in both the clinical, laboratory, and research environments. She has recently worked on a project that has significantly improved testing for LNP patients, and she's going to share that with you today. The title of her talk is Plasma Cell Enrichment for FISH Analysis of Multiple Myeloma. So take it away.
Thank you, Tracy. So sitting in the dark room of the fluorescence microscopy room within cytogenetics, periodically you will hear a technologist say, I think it's real. Do you think it's real? I think it's real. And this is a discussion between the first and second scorer. And this is a good image of a good example of the type of case that they're actually discussing. So here, the blue circles are actually nuclei, and we're counterstaining with DAPI. The red signals are hybridizing to FGFR3, and the green signals are hybridizing to IgH. And what we're looking for in this case is fusion or translocation between the FGFR3 gene region and the IgH region, because within multiple myeloma, so this is a slide from a newly diagnosed multiple myeloma patient, and we want to determine what genetic abnormalities are occurring in this patient, because it's very prognostic and makes a huge impact on the type of treatments they're going to give these patients. So we look at this case, we see one abnormal cell out of 32. We continue to score, and again, we only see two abnormal cells out of 100. A second scorer scores it, and they also find a low level of abnormal cells. So we suspect it's real, but because it's below our normal cutoff, we need to report it as negative. And this concerns us because we could potentially be reporting a false negative result. Now, when we look at these cells, we need to keep in mind, as in this May-Grunwald-Giemsa stain slide, that the slides that we are looking at, they're not all tumor slides. We culture a whole bone marrow sample. It's harvested, and it actually represents, all the cells within the bone marrow except the red blood cells. Now, when you look at this slide, I'm sure some of you have already picked out plasma cell. I know there's some cytology technologists here. So there is one plasma cell within this image. Okay, if you suspected this one, you are correct. But when a hematopathologist receives an aspirate, they do a 500 differential count, and they're looking for at least 10% plasma cells in order to diagnose multiple myeloma, which is a cancer of plasma cells. So when we go back to our slide, morphological identification is not possible, nor are we trained in morphological identification of these cells. So all cells look very much similar because we're only staining the nuclei. And so we score all cells. And as I suggested, this case could be reported as a false negative. Now, although 10% plasma cells has been confirmed by the hematopathologist, there are several pulls that are taken from a patient. So the case or the sample we receive, often it's labeled number 5 or 6, but we don't know if it's the first pull as recommended in guidelines. Like for cytogenetics, for molecular risk stratification, we should be receiving the first aspirate, but we don't necessarily know which aspirate we're receiving. So thus, because of hemodilution and because of the focality of multiple myeloma disorders, we're never sure of what we're receiving. So for this reason, a few years ago, all three organizations, the Canadian College of Medical Genetics, the American College of Medical Genetics, and the European Consortium of Guidelines all recommended that we target the analysis of multiple myeloma in our assessment of the plasma cells. Now, multiple myeloma is a little different from other hematological disorders. There is considerable amount of focality, and that's why it's so important to target our analysis just to improve the sensitivity. So there's two ways we can do this. One is using immunofluorescence, and this is highlighting either the kappa or lambda light chains, which are known to be expressed. In most, it's like highly expressed, either kappa or lambda within plasma cell neoplasia. Or we can actually pull those cells out of the bone marrow, and this is using a cell surface cell surface receptor that's found on the plasma cell called CD138. And it's a very clever and simple technique to label these cells with a magnetic bead and use magnetism in order to pull those cells out of the bone marrow sample. So we actually tested both. And because of the potential to automate the cell sorting of plasma cells from bone marrows, we decided to go with that route, and just the ease for technologists when they're scoring cells at the microscope. It's much easier to score all cells as opposed to making a decision which cell is, has good immunofluorescence versus not. And we did find some inconsistencies of our staining with the immunofluorescence technique. Now, Shab did go through this, but I just thought I'd quickly go through fluorescence in situ hybridization, because all of our testing for multiple myeloma is done using this very simple and clever technology. Simply, you have a DNA probe, which is an exact clone of the region that you're interested in, within the genome. We label it with a fluorochrome. There's 4 colors we usually use: red, green, yellow, and orange, and aqua. You hybridize using like heat and chemicals and pH. You can denature both your probe and your target DNA, and these are usually done on slides. So either actively dividing cells we find in bone marrows or bloods, and thus you can look at metaphases, or within archived paraffin-embedded tissue, cytology samples, or even our fixed bone or blood interphase cells. So there's 2 entities of DNA, either condensed or uncoiled, that we can look at and localize the DNA. And all of in situ hybridization depends on the remarkable stability of the DNA molecule. So DNA strand always binds to its complementary strand, and this is done because there's 2 hydrogen bonds between thymidine and adenine versus 3 hydrogen bonds between cytosine and guanine. And this allows us to, exploit the complementariness of DNA in order to hybridize other probes to it. And a good analogy for fish is essentially you're looking for a needle in a haystack, and that haystack can either be chromosomes or it can be uncoiled DNA of an interphase cell. And very simply, we can locate that needle in a haystack using like a DNA probe that's like a magnet. So it's very simple. But just as I discussed, we either use bloods or bone marrow, cytology samples, or paraffin-embedded tissue. We can fish anything and everything to localize that DNA. And we use it to enumerate the number of chromosomes, and intuitively you would think any chromosomal abnormality would have a delirious effect. However, within cancer, having this extra chromosome 8, which is found in many hematological disorders, 90% of myeloid lymphomas and 10% of lymphoid, it's actually not a bad, it's a fairly good prognosis for these patients. as well as 13q deletions. So we see these in about 50% of chronic lymphocytic leukemias, as well as 50% of multiple myeloma. And again, it's considered a intermediate prognostic factor, as there are other genetic abnormalities that provide a poorer prognosis for the patient. And this, I think, is such a clever and easy design. The majority of mutations within hematological disorders are actually translocations. And this simple technology to detect a translocation is just hybridizing two probes on either side of a breakpoint. One is labeled with red, the other labeled with green probe. And you can quickly visually detect whether or not a translocation has occurred. If they're overlapping, you get a nice fusion signal. And if you see a single red and a single green, a translocation has occurred because they're now separated. And another, like dual color, dual fusion, you label either gene that's involved in a translocation with either red or green. And if you get a fused signal, either like a red and a green very close to each other, less than a signal distance apart, or you get a nice yellow signal, you know that a translocation has occurred. Amplification, we use this in breast cancer, either low-level amplification or co-amplification of both the gene and the centromere region of chromosome 17, as well as like high amplification. But all of this, the FISH is very valuable in localizing DNA in its native context. Pathology is, like all of pathology, is based on in situ, or a lot of it is based on in situ analysis. So fluorescence in situ hybridization allows you to detect the abnormalities within its context. And yesterday, Melanie was talking about microenvironments and these large sections that they can take in order to look at the big picture. And in certain abnormality or certain cancers, that's really important to look at the big picture. So this is this happens within certain tumors, genetic abnormalities. You can see subclones and heterogeneity. So FISH is very valuable. In that sense, we validate all our probes on metaphases, and we do an extensive amount of validation using normal cells of the tissue that we're interested in. And this is to detect any abnormal patterns due to tissue artifacts, the process artifacts, overlapping cells. Is very important because all of laboratory medicine is based on establishing normal cutoffs. You need good cutoffs because you do not want to be reporting false positives. But again, we do not want to report false negatives. So it's very important to establish these cutoffs, and we do an extensive amount of work in that before any probe or test is brought to clinical practice. So now to talk about the exciting project I've been working on. Just a quick review of multiple myeloma. It is a cancer within the bone marrow, like plasma cells in particular. Normal, healthy plasma cells are responsible for producing many different isotypes of antibodies needed to fight many different infections. However, in multiple myeloma, there's a clonal growth, uncontrollable growth of the plasma cells, and thus they produce one type of antibody. And unfortunately, these monoclonal antibodies, or M proteins that they're called, are not functional, and these patients are not able to fight a lot of infections. And just this overproduction of these M proteins or abnormal proteins, there's overproduction of light chains as well, causes all kinds of havoc to the body. Not only the growth of abnormal cells crowds out the ability of other cells to grow well, so many of these patients have anemia. Just the accumulation of these proteins in the kidney causes can cause clogs, can cause all kinds of problems for the patient. But Here's just a quick summary, but some of these are not actually, these manifestations, clinical symptoms, are not always shown until there's a substantial amount of plasma growth. So as I discussed before, at least 10% plasma cells, are required, with monoclonal or M protein spike in the patient, and restricted either kappa or lambda light chains. And recently they've updated the criteria to diagnose multiple myeloma with before you had to have all the clinical indications. Now they appreciate if there's more than 60% of the bone marrow consists of plasma cells. That's indicative of further damage to come within the next 2 years. Monoclonal immunoglobulin ratio of greater than 100, and the presence of more than one focal lesion based on MRI. And these patients may or may not have clinical symptoms. So multiple myeloma develops, it's a multi-step development. But at the core of it, there's a primary genetic event, either an IgH translocation or hyperdiploidy. And this allows cells, it initiates the uncontrolled growth of cells. And there's two pre-malignant forms of multiple myeloma. So either monoclonal gammopathy of undetermined significance, or MGUS, or smoldering multiple myeloma. And these patients don't necessarily show any clinical symptoms, but it's the tumor's brewing. It's starting to grow. And so we see those primary genetic events. Secondary genetic events allow different clones to grow, and this is when the cells grow more substantially. There's progression, and there's secondary abnormalities. So within the lab, we want to detect these. The IgH translocations, we also want to look at the copy number abnormalities. So this is significant, and it's important for the overall care of the patient. And here's just a quick summary of the co-occurring mutations that occur within multiple myeloma. All the circles represent different clones. So again, primary events, either hyperdiploidy, which is trisomies of certain chromosomes, and then you get translocations occurring. This allows the cell to start growing, and then secondary events occur. Many are copy number abnormalities, like p53 deletion, which is common in many cancers. We have 1q gain, which we look for in our lab. MYC translocations occur, but we're not currently testing for that. But the abnormalities we are looking for are the high-risk cytogenetic abnormalities, and these are, IgH translocation, p53 deletion, a duplication of 1q, and also a deletion of 1p. These are all very important for the oncologist to know and determine how to treat these patients. So this is just a quick summary: primary cytogenetic events versus secondary cytogenetic events, and further cytogenetic events that result in progression of multiple myeloma. So previously at UHN, when we received a bone marrow, the whole bone marrow would be cultured. We would harvest it and then fix it and archive, and just wait until we receive a hematopathologist report indicating that multiple myeloma, they suspect multiple myeloma is present, and to go ahead and do the FISH analysis. So before, we were just looking at 13q deletion, 2 of the IgH translocations, FGFR3 and MAF, and then p53, and then a report would be given. And again, these were like FGFR3, MAF, and p53 are high-risk cytogenetic abnormalities for the patient. Now, what we wanted to do was test in parallel splitting our bone marrow samples and seeing what percentage of abnormalities we're seeing within a cultured sample versus what we're seeing within an enriched sample. And is it necessary? Does it make a difference? So we used in magnetic cell purification. It's a very simple technique. All we're doing is labeling our cells with an antibody that's conjugated to a microbead. So CD138 is expressed on both normal plasma cells as well as abnormal cells, and it's not expected to hybridize to other cells within our whole bone marrow sample. Upon labeling, and this incubation is very short, 15 minutes. Once that's complete, we add a wash to our sample, centrifuge it, and then we're going to sort the cells by adding it to a column that's under a magnetic field, and the beads within the column actually magnify the magnetic field by 10,000. So this allows us to sort very small concentrations of plasma cells within our sample. So it's very clever, very simple method. So all cells that do not have the antibody flow right through the column, but all cells that are labeled are retained within the column. We remove the column from the magnetic field and simply elute the cells. So now we have a negative fraction and a positive fraction. And then we would fix our sample and just wait for the case to be activated. So in our study, we wanted to simply determine what the fold enrichment was. What is the effect of delays in processing? As we all appreciate, we don't necessarily receive our samples the same day, and we know that the CD138 can start shedding off the cells within 4 to 6 hours. So it's very important that we track that information. So if we don't get good results, we can follow up. And also, if we could use robotics. Now, one thing I didn't tell you about is when we receive our samples, actually the first time we were talking to the company, Meltenyi, we were telling them we really want to determine whether or not we could batch our samples, because just for workflow, this is going to be challenging. We're already quite busy, and how can we limit the amount of time we need to do this? Could we batch our samples? And so in discussions with them, we decided, and like technical support, I'm sure all of you are involved in a lot of development. Technical support from a company is incredible, and you learn a lot, and we worked very closely with them. And it was determined that we could put our samples upon receipt in RPMI media and store them in the fridge. So we tested same day magnetic cell separation versus up to 72 hours later. And again, that was great working with their team in order to complete this. So batch processing we found was very valuable in addition to the robotics. So my very first sample, I was so excited. After enriching it, I look at my positive fraction and there's a pellet. And, any of you dealing with centrifuge tubes, when you get a pellet, it's very exciting because it's worked, there's cells there. And we were able to bring it to flow cytometry. And Armand did a quick assessment of our enriched sample, and they had previously done flow cytometry on this, and they found 2% plasma cells. But in our enriched sample, they found 70% plasma cells. So we were very excited. And then when I did the testing on that sample, I found 62% of the cells had a deletion versus only 4% in our cultured sample. Now this you wonder, how could this be? Like the hematopathologist reported 50 to 60% plasma cells within the bone biopsy. Flow cytometry saw 2%. So we we suspect that the aspirate, and we only work with aspirates in hematology, or the hematopathologist will report either on a biopsy or an aspirate, and like the current guidelines recommend that they report the higher percentage of plasma cells. But again, because of the focality of multiple myeloma, that bone biopsy sample he or she is looking, right at that area versus we're receiving, an aspiration. So it may be hemodiluted, and you can, start justifying why the percentage we saw was so different. But if we take that sample and we enrich it, look at the difference it makes. 62% abnormal. My second sample, hematopathologist found 50% within a blood clot. Flow cytometry found 11%, and I found almost 95% of the cells had a 13q deletion, 75% had a p53 deletion, and this was seen within our cultured sample, and thus, the fold increase was substantially less. But it was very good results for first few cases. And here's just some examples. We found with our cells going through a column, I don't know if any of you work with purifying samples like through a column. 15? Okay, thanks. But if you purify your sample, you get rid of a lot of debris and background, and it substantially improved the quality of our analysis. So that was exciting to see. So that's the sample with 11% seen by flow cytometry. Here's another example of a 13q deletion we detected. However, this was not found within the culture. Here's the IgH FGFR3 translocation. This was detected within the culture, 6%. However, in the enriched portion, we did find 44.5 on the same day analysis, and actually 48.5 the second day, but the difference is not significant. But again, we're seeing exactly what we expected to see. So the value of this technology, is proving itself. 1q duplication is a high risk factor in these patients, and in the culture we did detect it, but only at 8% versus 80% in the magnetically cell sorted sample. So quite exciting data. So we did demonstrate fold enrichment in 31 FISH abnormal cases. And just so you know, we did over 100 magnetic cell separations in order to generate 31 abnormal cases that were informative, because that was the only way we could detect or determine, if there was a fold enrichment. And if you look at the yellow highlighted column, you can see there's a full range of enrichment from 2 all the way up to 20. But bottom line is it's making a significant difference. We see, like you would hope you get 95%, but we don't necessarily get that. We're also isolating normal plasma cells, so we don't expect them to have abnormal cells. Some of our genetic abnormalities are just in some of the clones, so they're secondary abnormalities, and you don't expect to see them in all of the cells. So that's why we have such a wide range, but very exciting data. And again, just showing you the difference between a biopsy versus an aspirate. And again, flow cytometry traditionally has an estimate of fewer cells than what the hematopathologist will report, even from the same aspirate. And that's just, unfortunate effect of the technology. Plasma cells are very fragile, so we can lose some of them in the processing for flow cytometry. So it's just all of this needs to be considered and taken in context. because we are limited by the type of samples that we have to work with. And, within cytogenetics, we're, making a huge, or improving our sensitivity significantly. So in summary, of the 31 samples, we found 22 abnormalities that were not detected by the unenriched fish. And all of these have been, like, amended report was sent out for these patients once we validated our technology. So 7 cases with 13Q, 10 cases with a duplicated 1Q, 1 case with an IgH MAF, and actually 1 case that had an FGFR3 IgH. It was considered a false positive, and this was, simply because of overlapping of signals. So we found that there were separate signals in the bone marrow sample. For our delay in processing, we were quite excited. We found 100% concordance all the way up to 72 hours. So we could batch our samples. We didn't need to do magnetic cell sorting on the weekend, as we could set it up on a Friday in media, put it in the fridge, and then on Monday perform the magnetic cell sorting. So we have a new paradigm at UHN. All bone marrow samples received require, assuming that the requisition is complete, clearly indicating that multiple myeloma testing is to be done. Those samples are put in media, put in the fridge, so that they can be medically cell sorted. We have 3 fish probes that we do initially. So IgH break apart, P53 deletion, and 1P, 1Q, and these are high-risk cytogenetic abnormalities. And then if the IgH break apart is proven to be positive, then those cases are reflex, because the oncogene partner in that translocation, the oncogene that's being turned on by the very IgH has high enhancers, strong enhancers within the gene. So when you translocate the gene to another oncogene, that oncogene is turned on, and that's what's fueling these tumors. So it's very important to find out which oncogene is turned on. And again, they all have different prognosis. So FGFR3 has a very high prognosis, very poor prognosis, whereas CCND1, the prognosis is a little more favorable. MAF also is very aggressive tumor. So all of this is very important to the clinicians. They rely on it for risk stratification. And finally, we looked at automation of the magnetic cell sorting. There was 2 platforms we looked at. These companies are amazing. They worked very well with us. Miltenyi with their AutoMax, as well as RoboCept technology. Miltenyi does use 2 columns, whereas RoboCept is column-free, which we like. And because then there's potential, lower potential of losing your cells. The cost was comparable, although it is a little more using automation, but then, you get the added time that you're going to save for your technologists. We saved processing for cases. We saved an hour. Fish results were comparable. However, we did find in the AutoMax that 1 case failed because there was too few cells. And in RoboCept, there was 1 case that failed because of the clumping. Pellet size overall, so for parallel testing of equal volumes, we found the pellet sizes were smaller. However, we were still able to achieve our results. But with working with stem cell, for the RoboCept sample, we were able to develop a customized SOP, and this significantly improved the retention of cells, because we were finding, I'll show you here, so here's manual, Miltenyi, and stem cell. But if you look at the tube, this is the original tube that went into the system, you can see there's residual red suspension in both the Miltenyi and the RoboCep, and this was residual volume, and we talked to both of them and said we need to address this because there is smaller pellets, and they both worked with me. Miltenyi, we recalibrated the aspiration arm, however it was unsuccessful, but with stem cell, they made many adjustments to the system, allowing like the aspiration arm just to sit on top of the tube to allow everything to drop back. They pre-wet like all the pipette tips before going into a solution to prevent any cells from adhering inside the pipette tip. Like all these little techniques that we do manually, we know to do that, like to limit loss of cells. But these weren't initially, programmed within these systems. But working with them, we were able to substantially improve the result. And this is just an example of the clumping, we found within RoboCep. And we found, actually, we've been testing the use of DNA. So when cells break down, free DNA is in the suspension. DNA attaching to cells can actually make cells very sticky. And if we added DNAs, it could help get rid of the DNA and thus prevent cells from sticking as much. And I've also read that some patients just have soluble like factors within their bone marrow that can also encourage this clumping together of cells. Plasma cells are very sticky, like to begin with. So we've done more washes, added DNAs, and so we're, improving this, our final result. So in the end, in conclusion, magnetic separation greatly improved the sensitivity for the detection of genetic abnormalities in multiple myeloma. Enrichment is robust and works even after 72-hour delay in processing. Sufficient plasma cells can be recovered from aspirates with very low levels of plasma cells. We had some samples with only 2% plasma cells. But the hematopathologist still wanted us to go ahead with the testing, so we did, and we were successful. We were able to achieve enough cells, and robotics will help improve testing and workflow. So I have many people to thank, oh my goodness, but certainly the Cytogenetics lab for working together with me and analyzing all these extra slides and cases and troubleshooting and all the fun stuff we love to do within the lab. I'm very fortunate working with a great team. I've had many discussions with Melanie from PRP, who has a wealth of knowledge, as I'm sure you all appreciate if you got to see her talk yesterday, working with immunofluorescence, flow cytometry, endless discussions about CD138 because they use that antibody and obviously for sorting out plasma cells and bone marrows. Miltenyi, biotech, the salespeople, the technical people from all the vendors that I've been fortunate to work with. And thank you. I'm sorry, I think I'm a little over time, but any questions?
I have a quick one. Are we offering, we're not offering this clinically yet, I'm assuming.
Oh, yes.
Oh, we are?
Yes, yes.
How long has it been?
Oh, gosh, probably a year and a half now.
Okay.
Yes, and we're still working on improving the robotic system. But yes, this is offered, and it's very exciting. It's great for the patients because we've significantly improved sensitivity. Working with the samples we receive, what can we do to ensure that we get an accurate result for the patient? Thank you.
Okay. Thank you very much.
You're welcome. Thank you.
Jeez, you're okay. Oh my God. Fell off the side. Good thing you didn't fall down. Here, this is for you.
Thank you.
Thank you
very much. Thank you.
Do you want to bring the raffle box up? So everyone has their tickets out. We'll get each of you guys to pull.
Here. All right. Here we go.
So our first number is 635927.
Yes!
All right. If you want to come up and grab your prize. There you go.
Oh, thank you.
And for the second $50 gift card, it's 635902. And thank you all for coming. Hopefully we'll see you tomorrow at 1 o'clock in Stella's. Good job.
Just under 1 o'clock.